Okay, let's look a little bit more at this antigen and antibody, the antibody and immunoglobulin stuff. So, in looking at what happens to activate the antibodies, okay? Because remember, with the B cells, we're either going to do the antibodies or those immunoglobulins. So the antibodies. Now, I'm going to start right here at the T cell, okay? Because the T cell, this is where we're going to have that antigen-presenting cell come in. So the APC, such as that macrophage, okay, that digested some of the pathogen, basically puts it on the plasma of its membrane. When it reaches the T cells, it will then activate that helper T cell. That helper T cell, because it was able to fit the antigen, it gets activated. Because we activated a T cell, the T cell can release the cytokines. That list that we looked at, okay? Now, the cytokines, those can activate the B cell. So the B cell got activated, started to proliferate, meaning increase in number, okay? Those B cells are going to, they might become a memory cell, and then we're going to have those plasma cells. And because this is antibody, it's secreting antibodies. So in that B cell, okay, if the B cell, if that antigen fits on the receptor, we've activated it, okay? Now, most of the time, we're going to have that T helper cell. Then we can move on to create antibodies so that we can attach to the antigen and basically like stop its activity. So in that antibody, that structure right there, this is what your textbook is going to kind of get a little detailed about. We just don't have that kind of time, okay? But at this point, all right, we can have some of these antibodies and these immunoglobulins, and it's going to be their job to work against something. Now, antibodies, they're going to be a protein that is termed the immunoglobulin. Basically, this globe, okay, this protein globe, it's going to have these chains. Like I said, your textbook's going to get a lot of detail. I don't want you to get into it. These are the immunoglobulins we have.
Do we need to get into these details?
Now, some of them a little more understanding than others, okay? But we have 5 immunoglobulins. G-A-M-D-E, Gamda. I'm not going to ask you to remember what each one of these does, but some are a little more common than others, a little more prevalent, okay? IgA, that'll be one that's rather prevalent that you'll hear. Bacteria and viruses, okay? Note what some of these other immunoglobulins will do. what they'll work against trying to invade the body. We can have it defend against bacteria, viruses, toxins. The complement, like I said, I'm not asking you to know all of them. IgA is a common one. IgG is one that's pretty prevalent. React with antigens, activate B cells, promote inflammation and allergic reactions. Now, here's what the antibodies can do. Okay, these immunoglobulins, this is what they can create in the body to try to fight something that's invading us. They can cause agglutination, precipitation, neutralization, opsonization, chemotaxis, agglutination, lysis, neutralization, inflammation. Depending upon the pathogen that has tried to invade us, okay, and the one that's going to get activated, okay, these are going to be able to have the ability to distinguish between those pathogens. They're going to be able to be specific, and this immune response that we can get, there's only one that gets to cross the placenta between mother and child for the immune response. That's going to be your IgG. IgG is going to have the ability to cross the placenta. Now remember, babies are born with a limited immune system. And then they'll have some immunity once they're born from the mom, okay? They'll pick up more if they're breastfed, but eventually all of that runs out and the child has to begin to develop their own immune system. So they're going to need to have the vaccinations, the exposure, that sort of thing. So IgG is going to be one that you will hear a little more about. So each one has the ability to go on and do something. Okay? They have the ability to make antigens, for example, clump up together. And if they can clump up together, that's going to make them inactive. So see how they might work to kill something off? They might cause it to lyse. They might cause that antigen to lyse, the cell to lyse. The job, be specific, go against a particular type of antigen and basically stop its activity. All right, so tomorrow we will finish this chapter and we'll start chapter 22. All right, so for today,